Mr. David is suffering from virus X for which a proper cure is not found yet. He is admitted to the hospital. The medical staff controls the illness using available medications. A week goes by, and Mr. David is now cured thanks to his immune system and the care from the medical staff. Doctors take a blood sample from David to make sure that his condition has returned to normal before discharging him from the hospital. The lab analysts then observe the blood sample under the microscope and extract the antibodies that have neutralized the pathogenic antigen. However, that antibody will not neutralize the other antigen variants taken from another patient. Therefore, they introduce different mutations to the extracted antibodies and check whether the modified antibody would neutralize other strains of antigen. The process is iterative, extremely labor-intensive, and consumes a lot of time. The process for a given antibody-antigen pair is extremely time-consuming as analysts need to come up with experiments to synthesize the proposed modifications to the antibodies, and need to repeat the experiment several times to obtain a mean value for the binding affinity of the given antibody-antigen variant. Accordingly, testing thousands of combinations seems almost infeasible and exhaustive. Nevertheless, this procedure has to be followed to design a suitable drug to negate virus X. The main issue with conventional experimental procedures is the time complexity and low throughput. However, with the proposed framework, we can test for the binding affinity of millions of antibody-antigen combinations in under 30 minutes. Further, our pipeline would automatically pick the best candidate antibodies for the antigen strains, which could then be tested experimentally. Accordingly, the screening process of monoclonal antibodies could be catapulted by integrating our system before lab experiments. That is, instead of conducting exhaustive experiments for all possible combinations of antibodies and antigens, we can now conduct experiments only for a handful of candidate combinations decided by our system. This could significantly reduce the time expended to develop a drug for virus X. With our model, the screening process was sped up and doctors were able to come up with an effective drug quickly. The vaccines were manufactured, tested, and mass distributed to be administered to the patients. The number of infected patients is going down and everyone is now immune to virus X. Everything goes back to normal. Now, we will gain some insights from a demonstration of the proposed system.